Hello students. So today we will be solving chapter 12 Respiration in Plants NCERT exercise. This is the disclaimer. Now let us take a look at learning objectives for our today's class. So we will explore and understand the fundamental concepts of respiration in plants by working through the NCERT exercise. This is the index. So we will solve question 1 to 12 of NCERT exercise for this chapter. So let's begin with the first question. So here we have to differentiate between question A, respiration and combustion, question B, glycolysis and Krebs cycle, and question C, aerobic respiration and fermentation. So let's look at the solution. So first let us start by differentiating between respiration and combustion. So respiration, it is a biochemical process. What does that mean? It means it involves chemical reactions within living organisms. Whereas combustion, it is a physiochemical process, which means it occurs outside of the living cells. Next point, respiration, as I already said, it occurs in living cells. It is very important for energy production in the form of ATP. Combustion, on the other hand, it does not occur in living cells and it does not produce ATP. So in case of combustion, no ATP is generated. For the process of respiration, various different enzymes are required at various different steps. So there are various enzyme catalyzed reactions taking place. And in case of combustion, enzymes are not required. Now, respiration, it is a biologically controlled process because it is taking place in the living cells and therefore it is a biologically controlled process. Whereas combustion is not an controlled process, it is an uncontrolled process. That is, there is uncontrolled energy release in combustion. Next, we will differentiate between glycolysis and Krebs cycle. So if we talk about glycolysis, it is a linear pathway. That is, it's a process where there is incomplete oxidation of glucose. So it starts with glucose and the end product is pyruvic acid, and it is a linear pathway. Whereas if we talk about Krebs cycle, it is a cyclic pathway. Now glycolysis, it occurs in the cell cytoplasm, whereas the Krebs cycle occurs in mitochondrial matrix. That is, these are the locations where these processes are taking place. So the pyruvic acid or pyruvate that is produced in the cytoplasm after glycolysis, it enters the mitochondrial matrix where Krebs cycle takes place. Now, third point, glycolysis, it occurs in both aerobic and anaerobic respiration. That is, the process of glycolysis is common to both aerobic and anaerobic respiration. So there is incomplete oxidation of glucose to form pyruvic acid, and this is taking place in both of these aerobic and anaerobic organisms. That is conditions. If we talk about Krebs cycle, it occurs in aerobic respiration. That is, oxygen is very essential for the process. Now, in case of glycolysis, there is generation of 2 NADH2 and 2 ATP molecules on the breakdown of one glucose molecule. Whereas if we talk about the end products of Krebs cycle, 6 NADH2, 2 FADH2 and 2 ATP molecules are produced on the breakdown of 2 acetyl coenzyme A molecules. Now let us look at question C, which is where we differentiate between aerobic respiration and fermentation. So aerobic respiration, it is a condition, that is, it is a process which is taking place in presence of oxygen. So here oxygen is used for deriving energy. That is, for energy production, oxygen is very important. Because oxygen here is the final electron acceptor to produce water. If we talk about fermentation, it occurs in the absence of oxygen. So either of alcoholic fermentation or lactic acid fermentation takes place in absence of oxygen. Aerobic respiration, what is the location? It occurs in cytoplasm and mitochondria. That is, glycolysis takes place in cytoplasm, Krebs cycle in mitochondrial matrix, and then we have ETC and oxidative phosphorylation in inner mitochondrial membrane. If we talk about fermentation, the only process that is common for both aerobic respiration and fermentation is glycolysis, which takes place in the cytoplasm. If we talk about the end products of aerobic respiration, what are they? It is carbon dioxide and water. Whereas end product of fermentation includes ethyl alcohol and carbon dioxide in case of alcoholic fermentation, and lactic acid in the case of lactic acid fermentation. Now, complete oxidation of respiratory substrate, that is glucose, takes place in aerobic respiration. Whereas in case of fermentation, there is incomplete oxidation of glucose or the respiratory substrate. In case of aerobic respiration, about 36 ATP molecules are produced. And if we talk about fermentation, only 2 ATP molecules which come from glycolysis are produced. Now students, let's look at the next question. What are respiratory substrates? And name the most common respiratory substrate. So let's look at the answer. So respiratory substrates, they are compounds that are oxidized during the process of respiration. For example, glucose. Like carbohydrates, especially glucose, acts as a respiratory substrate, which we have studied in glycolysis. There are other respiratory substrates like fats, proteins, and organic acids. Now let's look at the next question. Here we have to give a schematic representation of glycolysis. So glycolysis is the first step in breaking down glucose to extract energy for cellular metabolism. Here we can see the steps of glycolysis are given. So first, glucose is converted to glucose 6-phosphate, and in this process, ATP is utilized, producing ADP and inorganic phosphates. Then glucose 6-phosphate is converted to fructose 6-phosphate. The next step is where fructose 6-phosphate is phosphorylated and we get fructose 1,6-bisphosphate. In this step, ATP is utilized. So it is broken down to give ADP and inorganic phosphates. In the next step, fructose 1,6-bisphosphate is converted to triose phosphate. That is glyceraldehyde 3-phosphate and triose phosphate, that is dihydroxyacetone phosphate. So both of these are 3-carbon compounds. In the next step, glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglyceric acid. In this process, NAD+ is converted to NADH plus H+. In the next step, 1,3-bisphosphoglyceric acid is converted to 3-phosphoglyceric acid. Now this is the step where ATP is produced from ADP and inorganic phosphates. So now we have 2 molecules of 3-phosphoglyceric acid, and then they are converted to phosphoglycerate, that is 2-phosphoglycerate. In the next step, phosphoenol pyruvate is formed, and followed by this pyruvic acid is formed. So at the end of one glycolysis cycle, from one glucose molecule, we have 2-pyruvic acid, which is a 3-carbon compound. So it is important to remember to practice this cycle so as to remember the steps correctly. So the pyruvate and NADH, they are used in next stages of cellular respiration. Now let's look at the next question. What are the main steps in aerobic respiration and where does it take place? So let's look at the solution. So the major steps in aerobic respiration and the sites where they occur, they are listed in the given table. So in this table, we can see the steps of aerobic respiration and sites of occurrence. First, if we talk about glycolysis, it is the first step in aerobic respiration and it takes place in cytoplasm of the cell. The second step is Krebs cycle, which takes place in the mitochondrial matrix. The third step is electron transport chain, which takes place in inner mitochondrial membrane, where the electrons are transferred from one complex to another. And the fourth step, that is oxidative phosphorylation, it takes place through the F0, F1 particles in the inner mitochondrial membrane. That is, there is breakdown of the hydrogen gradient. So when there is breakdown of this gradient, the energy released, it derives the process of ATP synthesis. Now let us look at the next question. Give the schematic representation of an overall view of Krebs cycle. So here in the solution, we can see Krebs cycle is given, or the citric acid cycle. So in this step, the pyruvate, it is first converted to acetyl coenzyme A by oxidative decarboxylation. Then in the Krebs cycle, the acetyl coenzyme A, it is condensed with oxaloacetic acid and water to form citric acid. Acetyl coenzyme A is a 2-carbon compound and oxaloacetic acid is a 4-carbon compound. And together they form citric acid, which is 6-carbon. This citric acid is then converted to alpha-ketogluteric acid. In this process, carbon dioxide is released and NAD plus is converted to NADH plus H plus. Then this alpha-ketogluteric acid, which is a 5-carbon compound, there is decarboxylation steps. And after these two decarboxylation steps, there is formation of succinic acid, which is a 4-carbon compound. So here carbon dioxide is released, as well as NAD plus is converted to NADH plus H plus. There is also production of GTP from GDP. Succinic acid is then converted to malic acid, which is a 4-carbon compound, and during this process, FAD plus is converted to FADH2. Followed by this, what happens? Malic acid, it is converted back to oxaloacetic acid. And in this process, NAD plus is converted to NADH plus H plus. And then the cycle continues. Oxaloacetic acid is regenerated. So then again, it is condensed with acetyl coenzyme A to produce citric acid. Now let us look at the next question. Here we have to explain electron transport chain. So let's look at the solution. First, let's talk about the location and function of ETS. So electron transport system, it is found in inner mitochondrial membrane of the cell. Its primary role is energy extraction from NADH plus H plus and FADH2. So here in the image, we can see how energy is transferred or extracted from NADH plus H plus and FADH2. and these electrons are then transported from one complex to another. So let us look at the components and the flow of electrons. So the first complex is complex 1, which is NADH dehydrogenase. NADH dehydrogenase, in this complex, it oxidizes NADH plus H plus from glycolysis and citric acid cycle. It then transfers the electrons via FMN to ubiquinone. Ubiquinone, it is a mobile electron carrier. So here we can see that in complex 1, that is NADH dehydrogenase, the electrons from NADH plus H plus, they are transferred via FMN to the ubiquinone. Ubiquinone will further carry these electrons to the next complex, which is complex 3. The second complex is FADH2. So here there is transfer of electrons from FADH2 to ubiquinone. So here ubiquinone is further reduced by electrons from FADH2. This is complex 2, which is succinate dehydrogenase. So electrons from FADH2, they are then transferred to ubiquinone. The next complex is complex 3, which is cytochrome BC1. Now this complex receives electrons from ubiquinone. So ubiquinone, it transferred electrons from complex 1 to complex 3. And then it transfers these electrons, that is ubiquinone will now transfer these electrons to cytochrome C. Cytochrome C is another mobile electron carrier on the outside of this membrane. And this will further carry the electrons from complex 3 to complex 4. The next complex is complex 4, which is cytochrome oxidase. Cytochrome oxidase, this complex contains cytochrome A and A3 with copper centers. And it receives electrons via a mobile electron carrier, that is cytochrome C. So here we can see electrons from NADH plus H plus, they are first carried from complex 1, that is NADH dehydrogenase, to complex 3 via ubiquinone, which is a mobile electron carrier. Ubiquinone also receives reduced equivalents from complex 2, that is electrons from FADH2. Ubiquinone then passes on electrons to cytochrome C, which is another mobile electron carrier. Cytochrome C carries the electrons from complex 3 to complex 4. The next complex is complex 5, which is ATP synthase. So this complex is where there is ATP production during electron transfer. If we look at the energy yield, from NADH oxidation we get 2 ATP, from FADH2 oxidation we get 3 ATP. So this is complex 5, that is ATP synthase. So whenever electrons are passing through the electron transport chain, a gradient is developed. And when this hydrogen ion gradient breaks, the energy that is released is used to drive the process of ATP synthesis. Now let us look at the next question. Distinguish between the following. Option A, question A, aerobic respiration and anaerobic respiration. Question B, glycolysis and fermentation. Question C, glycolysis and citric acid cycle. So first we will distinguish between aerobic respiration and anaerobic respiration. So aerobic respiration, as the name suggests, requires oxygen. Anaerobic respiration occurs in the absence of oxygen. Aerobic respiration occurs in cytoplasm and mitochondria. That is, we know there is one process, glycolysis, which is common to both aerobic and anaerobic respiration, that takes place in the cytoplasm. After that, the end products of glycolysis, that is pyruvic acid, they are transported to mitochondrial matrix. Therefore, for aerobic respiration, the extra steps are Krebs cycle and ETS chain, and as well as oxidative phosphorylation, which takes place in mitochondria. Therefore, aerobic respiration, all of the processes takes place in cytoplasm and mitochondria. If we talk about anaerobic respiration, there is only one process, that is glycolysis, which is common to both, and it is taking place in the cytoplasm. The other processes, that is fermentation, in case of anaerobic respiration, also takes place in the cytoplasm. Next, what are the end products of aerobic respiration? Carbon dioxide and water. If we talk about the end products of fermentation, it is ethyl alcohol and carbon dioxide in case of alcoholic fermentation, and lactic acid in the case of lactic acid fermentation. Both of these processes takes place in the absence of oxygen. In case of aerobic respiration, there is complete oxidation of respiratory substrate. That is, if we talk about glucose, there is complete oxidation to produce carbon dioxide and water, whereas in case of anaerobic respiration, incomplete oxidation of the respiratory substrate takes place. That is, after glycolysis, that is during glycolysis, we know there is incomplete oxidation of glucose, which gives us pyruvic acid. In case of anaerobic respiration, this pyruvic acid is either converted to alcohol and carbon dioxide or to lactic acid. So in both the cases, there is incomplete oxidation of glucose or carbohydrate taking place. Now, if we talk about aerobic respiration, 36 to 38 ATP molecules are produced, meaning that it is more efficient in energy production. In case of anaerobic respiration, only 2 ATP molecules are produced, which comes from the process of glycolysis. And therefore, it is much less efficient in energy production. Now, let us differentiate between glycolysis and fermentation. Glycolysis, it is a process common to both aerobic and anaerobic respiration. And fermentation, it is a type of anaerobic respiration. So, in fermentation, glycolysis takes place. Second point, the end product of glycolysis is pyruvic acid, whereas the end product of fermentation is either ethanol and carbon dioxide in case of alcoholic fermentation, or lactic acid in case of lactic acid fermentation is the end product. Next, we will differentiate between glycolysis and citric acid cycle. Now, glycolysis, it is a linear pathway starting from glucose to the end product pyruvic acid. Whereas citric acid cycle, which is Krebs cycle, it is a cyclic pathway. That is, in case of Krebs cycle, oxaloacetic acid is regenerated, which then undergoes condensation again with coenzyme A, that is acetyl coenzyme A, to produce citric acid. Glycolysis, it is a process which occurs in cell cytoplasm, and Krebs cycle occurs in mitochondrial matrix. So first, glycolysis takes place, end product is pyruvic acid. This occurs in the cytoplasm. Now this pyruvic acid is then transported to the mitochondrial matrix where the Krebs cycle takes place. Now, glycolysis is a process which is common to both aerobic and anaerobic respiration. That is, for both aerobic and anaerobic respiration, the first step is glycolysis, that is formation of pyruvic acid. In case of citric acid cycle, it occurs only in aerobic respiration. That is, this process occurs in the presence of oxygen. What are the end products of glycolysis? One glucose molecule breaks down to generate 2 NADH2 and 2 ATP molecules. So these are the end products of glycolysis. If we talk about the Krebs cycle, it produces 6 NADH2, 2 FADH2 and 2 ATP molecules on the breakdown of 2 acetyl coenzyme A molecules. So this is a more energy efficient step, that is, more energy is produced in the Krebs cycle as compared to glycolysis. Now let's look at the next question. What are the assumptions made during the calculation of net gain of ATP? Let's look at the solution. So for the theoretical calculation of ATP molecules, there are various assumptions that were made. So let us look at those assumptions. First, so it is assumed that various parts of aerobic respiration like glycolysis, that is the various processes in aerobic respiration, glycolysis, TCA cycle or Krebs cycle, and the electron transport system, they occur in a sequential and orderly pathway. This is the first assumption. Second assumption, during the process of glycolysis, the NADH that is produced, it enters into mitochondria to undergo oxidative phosphorylation. The next assumption is that glucose molecule, it is assumed to be the only substrate, while it is assumed that no other molecule will enter the pathway at intermediate stages. So the only substrate entering the cycle is glucose molecule, and there is no other molecule that will enter at any other intermediate stages. And the fourth assumption is that the intermediates that are produced during respiration, they will not be utilized in any other process. So these are the assumptions that are made for theoretical calculation of ATP molecules. Now let us look at the next question. Discuss the respiratory pathway is an amphibolic pathway. So let us look at the solution. Now the process of respiration, it is generally considered or assumed to be a catabolic process. Why? Because during respiration, various substrates are broken down for producing energy. Carbohydrates, they are broken down to glucose before entering respiratory pathways. If fats, then fats, they will first get converted to fatty acids and glycerol, whereas fatty acids, they get converted into acetyl coenzyme A before entering respiration process. And in a similar manner, if we talk about proteins, they are also first broken down into amino acids, which will enter the process after deamination. Now, during synthesis of fatty acids, acetyl coenzyme A is withdrawn from the respiratory pathway. And for synthesis of proteins also, respiratory substrates get withdrawn. And therefore, in respiration, we also see synthesis, which is also called anabolism. So here we can see that because in the respiratory pathway, there is breakdown of molecules as well as synthesis of molecules. Therefore, we call it an amphibolic pathway. So during catabolism, larger molecules are broken down into simpler ones. They enter the respiratory pathway. And in the anabolic part, if we see, molecules, that is the smaller molecules, they build up together to form larger molecules with the input of energy. And therefore, both anabolism and catabolism, they are involved in respiration, and therefore respiratory pathway is an amphibolic pathway. Now let us look at the next question. So define respiratory quotient and what is its value for fats. Let's look at the solution. What is respiratory quotient? So respiratory quotient or respiratory ratio, it can be defined as the ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed during respiration. That is, carbon dioxide evolved divided by oxygen consumed is the respiratory quotient. Now, this value of respiratory quotient, it depends upon the type of respiratory substrate. For example, the respiratory quotient value is 1 for carbohydrates. However, it is always less than 1 for fats. Why? Because fats consume more oxygen for respiration as compared to carbohydrates. It can be illustrated through the example of tripalmitic fatty acid, which consumes 145 molecules of oxygen for respiration, whereas 102 molecules of carbon dioxide are evolved. So what will be the respiratory quotient value for tripalmitin? It will be 0.7, because it is the ratio of carbon dioxide evolved divided by oxygen consumed. So we have 102 divided by 145, which gives us value of 0.7. So respiratory quotient value for fats is normally around 0.7. Now let us look at the next question. What is oxidative phosphorylation? Let's look at the solution. Oxidative phosphorylation, it is a process in which electrons are transferred from electron donors to oxygen. Oxygen is the ultimate electron acceptor. The oxidation reduction reactions, they are involved in the formation of a proton gradient. So here a proton gradient is generated, which later drives ATP synthesis. Now the main role in oxidative phosphorylation is played by the enzyme ATP synthase, which is also complex 5 of the electron transport chain. This enzyme complex, that is ATP synthase, it has two parts. That is, it has two components, F0 and F1. The F1 component or the F1 headpiece, it is a peripheral membrane protein complex. And this contains the site for ATP synthesis. ATP is synthesized from ADP and inorganic phosphates. Now if we talk about F0, this component, it is part of membrane protein complex and it acts as a channel for crossing of protons from inner mitochondrial membrane to the mitochondrial matrix. So basically when this proton gradient breaks, the energy that is released, it actually drives ATP synthesis. So these protons, they move from inner mitochondrial membrane to the mitochondrial matrix. So for every 2 protons that are passing through the F0-F1 complex, there is synthesis of 1 ATP molecule. Students, now let us look at the next question. What is the significance of stepwise release of energy in respiration? Let us look at the solution. Now the process of aerobic respiration we have already studied is divided into 4 phases. First is glycolysis taking place in cytoplasm, followed by the tricarboxylic acid cycle or the Krebs cycle. Then we have the electron transport system. Then finally we have oxidative phosphorylation where ATP is produced. Now it is generally assumed that the process of respiration and production of ATP in each phase, it takes place in a stepwise manner. The product of one pathway forms the substrate of the other pathway. That is, for example, from glycolysis, the end product of pyruvic acid. This pyruvic acid undergoes oxidative decarboxylation. We get acetyl coenzyme A, which is the beginning of the tricarboxylic acid cycle. So here we can see end product of one process is the starting product of another. That is the substrate of the other pathway. Now, various molecules produced during respiration, they are involved in other biochemical processes. So the respiratory substrates, they enter and they withdraw from pathway on necessity. That is, as and when required, These respiratory substrates, they can enter a pathway or they can move out from a pathway. ATP is utilized wherever required, and the enzymatic rates, that is the enzyme catalyzed reactions, their rates are generally controlled. And therefore we see that there is a stepwise release of energy, which makes the system more efficient in extracting and storing energy. So this was all for today's class. Thank you.